Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like and subscribe the channel. In the last session, we studied about passive transport, active transport, and pumps. In today's session, we will continue further. Clinical applications of channels. Sodium channel. Local anesthetics such as procaine act on sodium channel both as blockers and on gating mechanisms to hold the channel in an inactivated state. Point mutation in sodium channel leads to myotonia, characterized by increased muscle excitability and contractility. In Liddle's disease, the sodium channel in the renal epithelium are mutated, resulting in excessive sodium reabsorption, water retention, and elevated blood pressure. Potassium channel mutations in long QT syndrome leads to inherited cardiac arrhythmias where repolarization of the ventricle is delayed, resulting in prolonged QT intervals in ECG. In this diagram, we can see that decreased potassium in myocytic cells leads to prolonged action potential and increased QT syndrome. Chloride channel. The role of GABA and glycine in inhibitory neurotransmitters is attributed to their ability to open the chloride channels at the postsynaptic membranes. In this diagram, we can see that strychnine and glycine are inhibitory to chloride channels. Cystic fibrosis is due to certain mutations in the CFTR gene, that is, cystic fibrosis transmembrane regulator protein, which is a chloride transporter. Retina. The excitation of retinal rods by a photon is by closing of action specific channels, resulting in hyperpolarization of the rod cell membrane. This light induced hyperpolarization is the major event in visual excitation. In this diagram, we can see that cystic fibrosis, there is a mucous membrane due to mutant CFTR gene. Voltage gated channels. These are opened by membrane depolarization. The channel is usually closed in the ground state. The membrane potential change, that is voltage difference, switches the ion channel to open, lasting less than 25 milliseconds. In voltage gated channels, the channels open or close in response to changes in membrane potential. In this diagram, we can see that in the resting state, sodium level is greater in exterior, whereas sodium channels open up and sodium channels are increased intracellularly. They pass from closed to open channel to inactivated state of depolarization. Once in the inactivated state, the channel cannot reopen until it has been reprimed by repolarization of the membrane. The voltage gated sodium channels and voltage gated potassium channels are the common examples. In the diagram, we can see a voltage gated sodium channel. These are seen in nerve cells and are involved in conduction of nerve impulses. Ion channels allow passage of molecules in accordance with concentration gradient. Ion pumps can transport molecules against the gradient. Ionophores. They are membrane shuttles for specific ion. They transport antibiotics. Ionophores increase the permeability of membrane to ions by acting as channel formers. In this diagram, we can see that an ionophore is a bacterial cell through which sodium ions pass. The two types of ionophores are mobile ion carriers, that is, example valinomycin, and channel formers, example gramicidin. They are produced by certain microorganisms and are used as antibiotics. In this diagram, we can see that an ionophore made from valinomycin, that is an antibiotic which allows passing of potassium ions. In this diagram, we can see that an ionophore made from gramicidin, which again is an antibiotic. When cells of higher organisms are exposed to ionophores, the ion gradient is dissipated. Valinomycin allows potassium to permeate mitochondria and so it dissipates the proton gradient. Hence, it acts as an uncoupler of electron transport chain. Second, active transport. The salient features of active transport are: first, this form of transport requires energy. About 40% of the total energy expenditure in a cell is used for active transport system. Second, the active transport is unidirectional. In this diagram, we can see that active transport of sodium and potassium ions requires energy in the form of ATP. It requires specialized integral proteins called transporters. The transport system is saturated at higher concentrations of solutes. The transporters are susceptible to inhibition by specific organic or inorganic compounds. In this diagram, we can see that an integral membrane protein. Sodium pump. It is the best example for active transport. Cell has low intracellular sodium, but concentration of potassium inside the cell is very high. This is maintained by sodium potassium activated ATPase, generally called as sodium pump. The ATPase is an integral protein of the membrane. In this diagram, we can see that ATPase is an integral membrane protein. It has binding sites for ATP and sodium on the inner side and the potassium binding site is located outside the membrane. It is made up of two pairs of sodium unequal subunits, that is alpha 2 and beta 2. Both subunits of the pump, that is alpha and beta, span the whole thickness of membrane. Calcium pump. An ion ATP dependent calcium pump also functions to regulate the muscle contraction. The specialized membrane system called sarcoplasmic reticulum is found in skeletal muscles, which regulates the calcium concentration around muscle fibers. In the resting muscle, the concentration of calcium around the muscle fibers is low. But stimulation by a nerve impulse results in sudden release of large amounts of calcium. This would trigger the muscle contraction. The function of calcium pump is to remove cytosolic calcium and maintain low cytosolic concentration so that a muscle can receive the next signal. For each ATP hydrolyzed, two calcium ions are transported. In this diagram, we can see that calcium ions pass through the membrane and then they pass through membrane of sarcoplasmic reticulum. Uniport, symport, and antiport. Transport systems are classified as uniport, symport, and antiport systems. Uniport system carries single solute across the membrane. Example, glucose transporter in most of the cells. Calcium pump is another example. If the transfer of one molecule depends on simultaneous or sequential transport of another molecule, it is called as co-transport system. The active transport may be coupled with energy indirectly. Here, movement of the substance against the concentration gradient is coupled with movement of a second substance down the concentration gradient. A second molecule being already concentrated within the cell by an energy requiring process. The co-transport system may be either symport or antiport. The symport In symport, the transporter carries two solutes in the same direction across the membrane. Example, sodium dependent glucose transporter. Fluorescein is an inhibitor of sodium dependent cotransport of glucose, especially in the proximal convoluted tubules of kidney, produces renal damage and results in renal glycosuria. Amino acid transport is another example for symport. In the antiport system carries two solutes or ions in opposite direction. Example, sodium pump or chloride bicarbonate exchange in RBCs. Before moving further, I want to tell you that please like, share and subscribe the channel. Your one click on the subscribe button shall help me to motivate and prepare other videos. Also, any suggestions can be given in the commentary box. Coming to the clinical applications, in the Hartnup disease, the transport mechanisms for amino acids are defective in the intestine and renal tubules. In cystinuria, the renal reabsorption of the system is normal. Renal reabsorption of phosphate is decreased in vitamin D resistant rickets. Diseases due to abnormalities of transport systems include familial hypercholesterolemia, then cystic fibrosis, then congenital long QT syndrome, Wilson's disease, eye cell disease, hereditary spherocytosis, and paroxysmal nocturnal hemoglobinuria. Secretory vesicles and exocytosis. Under appropriate stimuli, the secretory vesicle or vacuole move towards and fuse with the plasma membrane. This movement is then created by cytoplasmic contractile elements and the microtubule system. In the diagram, we can see exocytosis. The inner membrane of the fuse vehicle fuses with the outer plasma membrane, while cytoplasmic side of vesicle fuses with cytoplasmic side of plasma membrane. Thus, the contents of vesicles are externalized. This is known as exocytosis or reverse pinocytosis. Release of trypsinogen by pancreatic acinar cells, then the release of insulin by beta cells of Langerhans, and release of acetylcholine by presynaptic cholinergic nerves are examples of exocytosis. Often, more hormones are the signal for exocytosis, which leads to calcium ion exchanges triggering the exocytosis. Endocytosis is the mechanism by which cells internalize the extracellular molecules to form an endocytic vesicles. This requires energy in the form of ATP as well as calcium ions in the extracellular fluid. Cytoplasmic contractile elements take part in this movement. In this diagram, we can see endocytosis. In general, plasma membrane is invaginated enclosing the matter. This forms the endocytic vesicle. The endocytosis may be pinocytosis or phagocytosis or receptor mediated endocytosis. Pinocytosis literally means drinking by the cell. Cells take up fluid by this method. This fluid phase, pinocytosis, is a non selective process. Whereas receptor mediated endocytosis, the selective or absorptive pinocytosis is receptor mediated, also called as absorptive pinocytosis. Example, low density lipoprotein, that is LDN, is a good example. LDL binds to the LDL receptor and the complex is internalized. The sodium cytoplasmic side of these vesicles are coated with filaments mainly composed of clathrin. These are clathrin coated plates. After the LDL receptor complex is internalized, the receptor molecules are released back to the cell surface, but the cell LDN is degraded by lysosomal enzymes. Several hormones are also taken up by the cells by receptor mediated mechanisms. Here, here in this diagram, we can see clathrin dependent endocytosis, and there are clathrin coated vesicles. The protein dynamin, which has GTPase activity, is necessary for the internalization of clathrin coated bits. Many viruses get attached to their receptor specific receptors on the cell membrane. Examples are influenza virus, hepatitis B virus, polio virus, and HIV. They, they are taken up by caveole mediated processes. Caveole mediated endocytosis is also known as photocytosis. In this diagram, we can see alveolar mediated endocytosis and dynamin required by internalization. Phagocytosis. The term is derived from the Greek word phagin, which means to eat. It is the engulfment of large particles such as bacteria by macrophages and granulocytes. They extend pseudopodia and surround the particles to form phagosomes. Phagosomes later fuse with lysosomes to form phagolysosomes, inside which the particles are digested. An active macrophage can ingest 25% of their volume per hour. In this process, 3% of plasma membrane is internalized per minute. The biochemical events accompanying phagocytosis is described as respiratory burst. In this diagram, we can see that there is formation of phagolysosome. Here ends our session. Kindly like, share and subscribe the channel. Thank you.